Hello everyone, my name is Tomasz Kurowski. I work at Cranfield University, and thank you very much to the organizers for inviting me here today. So you may have noticed that the title of my talk is kind of general, talking about plant biology. It's a vague term. Maybe I could have been more specific and say plant breeding, for example. But really the idea here is that I feel, maybe I'm wrong, there is mostly focused on the biomed side of bioinformatics. Most people work in that. I myself started in medical biotechnology, but then I've transitioned into plants, into plant breeding, and I feel like I've taken it upon myself to represent that sort of field in that area of application. Bioinformatics, maybe some of you already work in this. Hopefully this will be interesting to all of you, and the idea is really not to focus on any single specific set of results, but to talk about the sort of challenges that you would face as a bioinformatician working with plant biologists and volunteers. But first, a couple of words on the institution I work at. So I work at Cranfield University in the UK. Our campus, a bird's view of it, bottom left here. So the idea, the core concept, so this is taken from a marketing blurb by our grant team. The idea is that Cranfield is a specialist university that mostly focuses on research, and we do only postgraduate courses. So we have master's students, we have PhD students, we don't have any undergraduates, thank God. And we are focused on a select number of fields that we are involved in through industry contacts. So basically we do a lot of consulting. Most of our projects are with external partners, so if a student works on a master's project, that's usually somehow connected with an external institution, a company. And mostly Cranfield is known for its aerospace field, so we have our own airport, but if you are already in a village like Cranfield, in the middle of nowhere, you also have space then for example, glass houses. And so this is our plant phenotyping glass house on the upper right here. Bonus points for anyone who happens to know why it's lit up in purple specifically. But specifically I work in the bioinformatics group at Cranfield, so don't only work with plants, I just happen to be focused on that. We work with omics tools for plant crops, yes, but also fungi, bacterial pathogens. We do a lot of machine learning and data science for personalized nutrition, for food authentication, for soil management, and we work with quite a few external collaborators, including the London School of Hygiene and Tropical Medicine, where I believe Agata did her master's project there, and she is actually one of the alumni of our master's in applied bioinformatics course. So that's an example of what we do. We work with external partners to do this sort of applied research, and the head of our group is Vadim Mohareb, this guy next to me right here. The group is quite small, not as small as in this picture. There's, I think, 15 of us at the moment, 4 academics and 10, 11 postdocs and PhD students, research assistants, and so on. And like I said, we run a master's in applied bioinformatics course. This is the longest running bioinformatics MSc course in the UK. Relatively small, it's a postgraduate course. We have around 20 students a year. And I just started we're very much focused on sort of NGS, genomics, quite a lot of programming is involved as well. So we do Java, for example, Python, and R. But leaving that aside, I was going to talk about plants and advocate for plant research, for bioinformatics and plant research. So I usually use 2 anecdotes to illustrate why working with plants is very rewarding and interesting. So one of them is focused on this sort of experimental setup that we have on the right here. I won't be going into too much detail, but the idea here is this is something called basically a pedigree chart. So it's describing what we do when we prepare our experiments. So what we've done here is we're working on tomato phenotyping here. We have one parent, female parent, Elsa Craig. It's a type of tomato. The other male parent is also another tomato. We cross those tomatoes. We have our F1 population, and then we basically cross this F1 population with itself to produce an F2 population of 100 individuals. So this X symbol that stands for something. So basically we've crossed 2 people. Yes, we've made a child, and then we've made a population of very inbred grandchildren. We've had some Biomed people at one of our seminars when we were showing something like this, and a certain professor nodded sagely at this and commented, "That's not what we do with humans." So the idea is that you have a lot of freedom in terms of experimental setup when you work with plants. There's a reason why the history of genetics really starts with Mendel and peas. Plants tolerate inbreeding very well, so you can get a slightly simpler genetic scenario and background in your experiments. Generations go by fast, and there are fewer ethical concerns, you could say, in general. You can set up your experiment however you like. And also, yes, some people work with model plants like Arabidopsis, but you can also do very applied research in this. So this is from a project called Genetic Control of Nutritious Rooting in Horticultural Crops. And this was in collaboration with Syngenta, so that's a huge company, and Edward Vincent Limited, that's a much smaller plant breeder focused on soft fruit. So basically, the research that we deliver to them, that will go directly into producing seeds, for example, that will be sold directly to farmers and then directly to the consumer. So if you work in biomed, you know that if you discover a cure for cancer tomorrow, it will take maybe a decade for it to actually go into clinical use. In plants, this turnaround is much faster, so you can make an impact in your lifetime, you could say, on a much smaller scale. So that's one example I usually give. The other one is also mostly intended as just a funny quip, an anecdote. So if you genotype an individual to establish what's the probability of developing a specific phenotype, in plant breeding, you would call that genetic potential. So you would say that you have a genetic potential of some percentages of developing large fruits. Yeah? In biomed, you would usually call that genetic risk. So this is kind of a completely different perspective of what you're working at. In plants, you usually try to improve things. You're trying to make a better, tastier fruit that stores better, survives transport, whatever else, whatever your founder happens to be interested in. In biomed, you usually try to prevent diseases. You try to basically, you're more defensive and less creative, you could say. So to summarize this, you could say that because I'm very impatient and because I'm a cowboy who likes to shoot from the hip, I moved to the countryside and started growing plants. So now for the sort of challenges that you may encounter while working with plants. So usually you're working with non-model organisms. So if you're working with human data, you have a very well-established human reference genome that thousands of people have worked on. In plants, you will usually have to maybe create your own reference assembly, or you will work with one which was generated relatively recently on a modest budget a few years ago, and then over the years, it may need to undergo some changes and updates. So these are eukaryotes. So plants are relatively big. It's not like authentic microbial genomes. It's not quick or cheap. Tomato, for example, I'm mostly working with tomatoes. That's 1 gigabase, so 1/3 of the human genome. And this is from the SCI Soil Genomics Network website. That's a group that basically maintains resources for tomato and related species. These are the updated versions of the tomato reference genome. So the one that was actually first released with the with the tomato genome paper in Nature, that was the 2.4 from July 2011, and since then we've had 3 additional updates. I also work with rosacea, I work with raspberry and strawberry, so that's another of these sort of research websites. But the scale is much smaller than in human. And something that you may encounter when working with something like this is the reference genome is something very basic, really, in most research. So it's meant to serve as a ostensibly static reference that you align things to, that you annotate. And that's a problem if it's a work in progress, as I mentioned in the previous slide, because if you've updated your genome, what happens to all this downstream data? Do you rerun everything from scratch? That would be the gold standard, really. Do you just move things around to update the coordinates? Some of you may remember when the latest version of the human reference genome came out, 38, I believe, is now. That was 6 years ago now. But people like the 1000 Genome Project, they did both of those things. First, they updated the coordinates by liftover, and then, because it takes more time, they actually redid all the variant calling to update the data. In tomato, they had to do that as well because they moved things around between 2.4 and 2.5. and actually they felt they were lucky because they only moved things around in terms of reorganizing the scaffolds. They didn't actually remove any sequence, they didn't add any new sequence, so in principle it should be enough just to move things around. And in 2015, when this happened, well, 2014, this happened, I started my PhD in 2015. So I started working on tomatoes, and my idea was, well, I want to figure out how they did this. And I was learning basics, and it's an advisable thing to do if you try to learn about something, to try to make your own version, just to figure out how everything works. Even if your version is rubbish, at least you've understood what's going on. And my problem was, I made my own version of this, and my results, my variants were completely different than what was on the official website. So I took a few weeks to figure out what am I doing wrong. So basically, this is the map, these are the positions within chromosome 11 in tomato. And all the variants I was getting here were correct, the same as theirs, and all the variants in here were wrong. It turned out they screwed it up. So basically, when they updated this, because they didn't have to do the lift, they didn't have to do the complicated lift over or rerun everything, they just had to move things around. They just updated the coordinates, but in places where they actually did inversions, so they inverted some scaffold, they forgot to do reverse complements for the bases. So all of the bases in areas where they flipped a scaffold turned out to be wrong, which explained why quite a few plant molecular biologists in our lab had trouble creating markers, PCR markers from those areas.
So how many years of research did they lose because of that?
Well, about a year. But yeah, we've tipped them off, they've updated, they fixed this. We were very happy. We've got some collaboration with them from that, so I was happy too. And it made for a nice chapter in my PhD thesis. And yeah, so basically they were updating data from this sort of paper that was resequencing a few hundred existing tomato lines, and those had to be remapped. And why we are developing all of these resources in the first place. So one thing that we do, and one that I'm focused on, is something called introgressive hybridization. If you think about it, when we've domesticated the tomato, for example, we've domesticated a particular tomato, and we've done a lot of inbreeding on it. We've basically limited the diversity of the genome. We have a limited amount of alleles actually available for plant breeding.